Okay, so we got to talk a little bit here about how the microscope is going to work. We talked about how to handle it yesterday. Obviously, you carry it with 2 hands. Okay, we talked about how the stage can move around and make that easy for you to see things without losing the focus on them. Okay, moving on to the lenses. Okay, there are 3 different lenses on your microscope. Okay, there's the scanning power lens. That's the short one with the red line on it. Okay, the scanning power lens magnifies 40 times, so it's only slightly better than Hooke's first microscope was. Okay, then you have the low power lens. That's the middle one with the yellow band. It magnifies 100 times. Okay, and then you have the high power lens. That's the longest one with the blue line on it. Okay, and that one can magnify 400 times. Okay, which one should you start with?
Lowest one?
The lowest one. Yeah, the scanning power lens. Okay, we want to make sure we're using the right terminology. Okay, the reason being is this. The scanning power lens is designed to do exactly as its name would imply. Scan the slide for the specimen. Okay, so when you look through the microscope, okay, no matter what setting you have it on, it looks kind of the same. There's a big lit circle. Okay, and usually in that lit circle, there's like a metal thing, okay, or it actually just looks kind of black, okay, in most microscopes, but it's actually a little piece of metal that does that, okay? That's the pointer. It indicates where the center of the field of view is. It's supposed to be there, okay? I only say that because I once had a group bring me 3 broken microscopes. Mr. Coderre, there's something wrong with this. Mr. Coderre, there's something wrong with it. I turned them on. I don't see anything wrong. What's wrong with it? Well, there's something in the middle. Is it a little black thing to point toward the middle? Yeah. The pointer. Okay, it's supposed to be there. Okay. So yeah, that's supposed to be there. It's really important actually because you always want to move the specimen to the center of the field of view before you change magnifications. Because on the scanning power, okay, and this is something you're going to want to write down in a minute here, okay, the scanning power, okay, magnifies 40 times. But the field of view, the diameter of that circle, is 4,000 micrometers. Okay, that little symbol there that I put before the M, that's mu. It is the Greek letter M, because obviously we can't use mm, that already stands for millimeters. Okay, we also can't use capital M, little m, because that stands for megameters, which is 1,000,000 meters. That's a lot different. Okay, a micrometer, okay, is, there are 1,000 micrometers in a millimeter, okay, to give you an idea. So when you look through the scanning power lens, the diameter of this is 4 millimeters. Okay? All right. So I don't see a lot of magnification, I don't see a lot of detail, but I see a lot of space. Makes it easy for me to find what it is I'm looking for. Because when I move up to the scan or to the low power lens, this is what's going to change. Because I'm magnifying more, I see less. Okay? The area that I can see shrinks. Okay? Because I'm blowing it up, so I don't see as much. That's why before I move up in magnification, I want to make sure that my specimen is on the end of the pointer. Because if I leave it out over here, I'm not going to see it when I move up to the higher magnification. Okay? Everyone kind of following me there? It's kind of like this. Let's say you're at the Saddle Dome. Okay, you're watching the game, and your friend texts you, and they say, "I'm at the game too." And you're like, "Oh, all right. Well, there's 17,000 people here, so I don't know, where you are." And then you say, like, "What section are you in?" "I don't know. Someone gave me the tickets. I don't know. But I'm wearing a bright yellow shirt." Okay, so if you want to find your friend, do you take out a pair of high-powered binoculars and start scanning each individual person in the crowd? I mean, you could do that, but it's going to take you a long time. Or could you just kind of look around with the naked eye and go, "Hey, there's a yellow shirt. Now I'll put up my binoculars and see if that yellow shirt is my friend." That's what you're doing with this. The scanning power lens is you looking around the stadium, okay, with just the naked eye looking for yellow shirts. Okay? Once you find a yellow shirt, then you increase the magnification. But do you see as many people through the binoculars? You don't. You see more detail. You can tell whether it's your friend or not, okay? But you don't see as many people. That's what we're doing with the scanning power lens, finding the specimen, and then increasing the magnification to see more and more detail. Okay? So when you go to the low power lens, okay, you will magnify 100 times, so more than 40. But when you magnify more, your field of view, okay, is reduced to 1,600 micrometers. Okay, so mu is basically a U with tails. Okay, if you're looking at how to draw it on Google Docs or any of the other word processor programs, you can just insert symbol, go to the Greek alphabet, and you'll see it there. Okay, it's mu. Now, before I move up to high power, obviously I'm going to want to center it again, because they do tend to move around a little bit when you change magnifications. Okay? But if I go up now to my high power lens, I'm still going to see that lit circle, but now whatever was in it is going to be really, really big because I'm seeing way less of the original circle I saw with the scanning power lens. Okay? With the high power lens, which is the blue one, okay, I magnify 400 times, which means I only see a field of view of 400 micrometers. Now you're probably thinking, okay, that's nice. Why is that important? Why do I have to know that? Knowing how this works is what enables us to estimate the size of something we would view through the microscope. If I'm looking at something on my high power lens, can I switch to another slide? Does anyone need this one anymore? So let's say that I switch to my high power lens. Okay, so I know that the distance from here to here is 400 micrometers. Okay, that's the field of view on my high power lens. So I'm looking at this cell. How big is it? Just an estimate.
50 micrometers? 50 micrometers?
50? Okay, maybe. I mean, so there's 1, that would be 2, 3, 4 and a bit. I'd say 50 might be on the small side, but it would be okay. Okay. All you do is just go, okay, so I know that's 400 micrometers. It takes up, somewhere between a fifth and a quarter of it. Just give me an estimate. Okay, I'd say 50 might be a little small, but I'd also say 200 would be way too big. Okay, it's an estimate. Okay, as long as it's reasonable, right? If you're looking at something like this and you tell me it's 1,200 micrometers, it's an estimate, but it's really wrong. Okay, I'd say I would probably accept, on this one, let's say 50 to 120 micrometers as a reasonable estimate for the size of that object. Okay, that's something that we have to be able to do in Science 10. Okay, is look through a microscope and go, this thing I'm looking at is approximately this big. Because that'll be important for our lab diagrams, which we'll talk about next week. Okay, how do we draw a proper lab diagram? Okay, questions on that? All right, so that's estimating the size of an object. Okay, that is also, okay. So maybe just copy down this little blurb here, okay, about estimating the size of the object, and make sure you have those field of view numbers. You probably have them already because you drew the picture I drew, okay, but make sure you have those numbers as well to help you with that. Okay, I'll give you a minute to copy that down. All right, I would like for you guys to copy down these 3 questions, okay? Give me some, some answers for those, just write down some answers for each one, okay? And then I'll give you about, maybe 5 or 10, and we'll go through those questions together, okay? It's just stuff from in that lesson. Okay, 3 points of the cell theory. Point number 1. Point number 1, guys.
All living things must have cells.
All living things must have cells or are made of cells, something about them all having cells, good, yeah. Okay, what's the second point? What do cells do?
The cell carries out basic functions of an organism.
Cell carries out basic functions of the organism. Third point?
New cells aren't created, they come from the division of other cells.
Exactly. Cells are not created, they come from the division of existing cells. As long as you know those 3 things, okay, that's super important for those. All right, who are the 4 people considered to be the most important in the development of the cell theory? What did they do? Okay, Luke.
Hooke was the first person to view cells.
Right, Hooke was the first person to view cells, also built the first microscope. All right, who's next? Claire?
Anton, he first viewed living cells and understood that cells are alive.
Right, okay, Anton von Leeuwenhoek. Okay, we always want to use last name, okay, if we're naming somebody. Okay, so last name is usually good enough. You don't necessarily have to know their first name. Okay, so Leeuwenhoek was the first person to view living cells. Okay, had that weird ping pong paddle shaped microscope that he had built. Okay, and then the 2 Swedish guys.
Cell theory?
Right, they came up with the cell theory. Their names were?
Schleiden and Schwann.
Right, Schleiden and Schwann. Okay, are the 2 Swedish guys who came up with the cell theory. Okay, so we've got those there, and then on what basis can we deduce that plants and animals may have had a common ancestor?
The cell composition.
Right, the structure of the cells have a lot of similarities, okay, and that's all we've looked at so far, but also their processes, okay, transport, metabolism, okay, other than their mode of nutrition, which is something we'll talk about today, they have a lot of similarities. Okay, other than how they get their sugar, once they've got their fuel, their sugar, everything is pretty much the same after that. Okay, all right. Any other questions on that? Okay, then we'll leave that one. Okay, and we'll move on to talking about when we do our lab diagrams. Okay, so I'm not going to do one for you today because it's been kind of glitching out on me for how I usually do it, so I got to play around with it here. But let's talk about the format of a lab diagram. Okay, you're not going to have to draw. Okay, like back in the day when I had to do lab diagrams, like in university, okay, we had to draw them by hand in pencil. My artistic ability is that of about a 2-year-old. Okay, when I draw people, their arms and legs come out of their heads. Okay, so I'm not good at drawing, and it was really a miserable experience. And on top of that, you couldn't shade. Okay, that was against the rules. You couldn't shade any part of your diagram. So if one part was darker, you had to stipple it, okay, more frequently than the other parts of it. Right? And so when everyone was doing their lab diagrams, it sounded like there was a whole flock of woodpeckers on crack in the lab, okay? Because everybody would be doing this and stippling their lab diagram. Okay, so we don't have to do that because why? Everybody has a digital camera pretty much at their disposal. Okay, so what we will do is when we're looking at things through the microscope, we'll take advantage of technology. Okay, because when I was your age, tablets were what you carved commandments into. Okay, so what you can do is you just take your camera and you put it right on the microscope, and it takes a little bit of fiddling around to kind of get it so you're looking down the lens, but then you can just take a picture of it, right? And then what we'll do is we'll insert that picture and draw labels on it and put our name and date and title and all that kind of stuff on it. That would be way better because what your camera says you were looking at is a far better representation of what you were looking at than what you could draw. Okay, would you agree? Okay. So when we're drawing lab diagrams, okay, what we want to do is have the picture, okay, and we want our lab diagram to take up a full page. Okay, one diagram per page. For format, your name and the date go in the top right corner. Okay, so whatever day it is we're doing that lab, or that you took that picture, okay, that's the date that goes there. And then, when you are labeling your specimen, okay, all of the labels go to one side, preferably the right, but there's not a hard and fast rule on that. Okay? What we don't want is for our specimen to look like it's exploding. Right? Because I get lab diagrams where somebody's drawn this and then the labels are going everywhere and it looks like it's exploding. Okay? It's really messy. It's hard. Okay? So all of the labels go to one side. And you'll notice all my labels end with a horizontal line, as should yours. Okay? If I have two structures like these ones here that are on the same level where it would be hard to put the two labels, you can go diagonally up or down and then over, okay, so that you still end in a horizontal line, but it looks neat and organized. Okay? Everybody with me on that? Okay, so that's how your labels should go, all to one side. Then, this is the weird thing, the title goes at the bottom. That's the thing people have the hardest time getting their head around, because titles are supposed to go on the top, but on lab diagrams they're supposed to go on the bottom. So all you do is put a title. This is an amoeba, so I wrote amoeba for the title, and the magnification you viewed it at, so I viewed this on high power, okay, and I estimated its size to be approximately 450 micrometers. All right, everybody with me there? And I'm okay, guys, like I just used a U here for mu. I'm okay if you do that too, rather than go looking for the mu symbol. It's a U, I know what you mean. Okay, put a U in there, it's fine. Okay? Questions on the lab diagram? Okay, you don't need any like special Photoshop kind of software or anything like that to do this. We're just going to use Google Draw. Hopefully it'll work. I was having a little bit of glitching with it today. But all you have to do is insert the picture into it, draw some lines on it, and put the text boxes in, and you've got your diagram. Okay, super easy stuff to do. All right, questions on that? So let's just write a couple of things down here about a lab diagram format. Okay, so let's just make a rectangle to represent a page. So one per page. Okay, so name, date, top right corner, okay, title, magnification, and size underneath. Okay, and labels all to one side. Okay, when we actually do the lab, I'll walk you through the process so that you can see it done and actually follow along on a Chromebook and do it yourself. Okay, so that you've seen it, so you'll see it. Anybody need that anymore? All right, let's, if you have your notes package, guys, you can go to lesson 4. That's where I'm gonna head to now. We're gonna talk about modes of nutrition, okay, and a little bit of structure stuff. Whoop, not the right one. Okay, I'll give you a minute here, guys, to find that. If you don't have your notes package, okay, maybe write down the key points here. I'll give you a minute on that, and then we'll talk about the rest here. So, what we want to talk about today is, like we talked about that the plants and the animal cells have a lot of similarities, okay? Today we're going to focus a little more on the differences, okay, and why the differences are present. Okay, the big reason that plant cells and animal cells look different is because of what we call their modes of nutrition. Where do they get the chemical energy that all organisms need to survive? Where do they get it from? Okay, what do plants do to get their energy?
Photosynthesis.
They carry out photosynthesis. Okay, and what do animals do? They eat. Yeah, they consume the organic material from other organisms as food. Okay, whether those organisms are alive or dead depends on the type of organism we're talking about. Okay, if it was like a scavenger, then it would be eating dead things, but they're still made of organic material that they can digest and convert to sugar. Okay, the modes of nutrition focus on where does the organism get its sugar from. Okay, the particular sugar is glucose. Okay? Everything you eat, if it's going to be converted to energy, has to be converted to glucose for your cells to use. All right? This is kind of why, like, if you're looking at, like, maybe, if you were trying to lose weight, you would cut certain things out of your diet and focus on other types of things. Like, if you wanted to lose weight, what's the first thing you take out of your diet or limit in your diet? Carbs, yeah. Why? Carbs are sugars. Carbs are most easily converted to glucose. Okay, so if you eat carbs, your body can turn those into glucose with virtually no use of energy, okay, and your body can use them as fuel. Okay, the second thing you cut out is probably fats. Right? Fats are the next easiest thing for your body to convert chemically to glucose. Okay, one of the hardest things for your body to convert to glucose is protein. Right? Which is why, lots of diets focus on eating more lean protein, or you have a protein shake, or things like that, because protein is harder for your body to break down. It actually has to invest energy in breaking proteins down and chemically converting them to fuel. Okay, now, be careful. Don't go in on fad diets. Okay, you can be happy with yourself regardless of how you are. Okay, but you have to be mindful, because sometimes you can do that really wrong. For example, there was this really fad diet years ago, and the guy who invented it, he died from kidney failure because he ate way too much protein and not enough of everything else. Okay, you got to be really careful. Everything in moderation is always a smarter idea than, hey, I got to do just this. I'm going to do the paleo diet or whatever. Okay, be careful, be smart, just eat healthy. Okay, all the food groups are important. Okay. So yeah, just kind of watch for that. But the reasoning behind that is some things are more easily converted to sugar than others. Okay? All right. We won't get to the cell diagrams today. We'll probably do that on Tuesday, but I have color handouts of the cell diagrams for everybody, so you don't have to worry about printing those off, and we'll go through them together. Okay, modes of nutrition. Those three words, super important. That's why there are differences between plant and animal cells. Okay, how they get their glucose is different. A plant has to have the structures necessary for carrying out photosynthesis and anything else that supports that process. Okay, whereas animals have to have structures within their cells capable of chemically converting whatever they eat into glucose. What part of a plant cell directly carries out photosynthesis? Anyone remember what it's called? Chloroplast, yeah. Okay. Chloroplasts are actually fairly large. Okay? When we do our lab where we're looking at plant and animal cells, you'll be able to see them. Okay? They're kind of a grayish green oval that is highly visible, and there's usually quite a few of them, okay, in each cell, so you'll be able to see them quite easily. They're fairly large. Okay. Now, plants also have a cell wall. Animals don't. What's the reasoning behind that?
Structure.
Yeah, I mean, plants don't do one thing that animals do. What is it? Move. Yeah. Okay. If you're a plant, you need to be rigid so that you can support the foliage, your leaves, that are trying to capture sunlight and carry out photosynthesis for you. Okay, so you have to have something that's rigid and strong to support that, right? Now, if plants could grow bones, yeah, certainly that could work, but the problem is that bones have connective tissue to other bones. That's what makes us capable of moving. Okay, animals don't have a cell wall because we need our cells to be flexible. Okay, because we're mobile, okay, everything's got to be able to move around without rupturing. Plants, they have some flexibility for like wind and things like that, but they're not mobile, so they don't have to have that measure of flexibility. Okay, so there's another kind of difference between the modes of nutrition. Okay, one stands still and the other doesn't. Okay, this isn't like the Lord of the Rings, right? Like Treebeard and the other Ents are not, wandering around and trying to, do things. Similarly, if you, as an animal, had cell walls, you'd look like you came straight out of Minecraft, right? Like, because you wouldn't be very flexible and you'd walk around all stiff, and okay, so that wouldn't work either. Okay, so first mode of nutrition we're going to look at are chemoheterotrophs. Okay, that would be anything that has to consume organic material from other organisms as a source of food. Okay, so from this one here, guys, I'd say like chemoheterotrophs means they must consume chemicals, organic chemicals, okay, from organisms to make energy, and their cells contain lysosomes that contain digestive enzymes to break down food. Okay, like if you're really trying to summarize that slide, that underlined stuff is the important stuff. Okay, so examples of chemoheterotrophs, or let's talk about the origin of the word. Okay, chemoheterotrophs. Trophs means feeding or feeder. Okay, it's what you eat. Okay, so chemoheterotroph means organic chemicals, chemo. Okay, hetero from other organisms to eat. So these are chemical other eaters. They are chemical other feeders. Chemoheterotroph means you eat other things as your food. Okay, you want to follow the origin of the word there? Okay, the other mode of nutrition that plants use is photoautotroph. Okay, so the trough part is still in there. The trough is the energy, what you eat. Okay, so photo means, well, it means light actually. The prefix photo means light, but yes, it does refer to photosynthesis. Photoauto means self, right? You're not using anyone else. Light self-feeding. Okay, so yeah, photo means, yes, I'm using photosynthesis. Light is my source of energy. I'm using it to make food for myself. Okay, photoautotroph. Okay. Now, these are very different organisms, would you agree? Like, they're not all animals. The one in the middle is a fungus. Okay, a fungus is the same mode of nutrition as an animal. It digests and eats the organic material from another organism, be it alive or dead. Okay? Some fungi are decomposers. Okay, so they will run their hyphae. Okay, the hyphae is the feeding mechanism. If you look at them under a microscope, they look like the roots of a plant. Okay, like just to look at, they have a similar structure, but they work a lot different. Okay, hyphae are first off microscopic. A whole bunch of hyphae will make a feeding mat called a mycelium. If you've ever watched The Last of Us, they use that term a lot. Okay, in The Last of Us, because it's the fungus that's turning people into zombies. Okay, and they use the term mycelium, and that mycelium is an area that the fungus is growing in, and anything that's there can be part of the feeding apparatus of the fungus. So it's actually not all that far off in terms of using the term correctly. Okay, the whole brain exposed zombies eating people thing, not so much. Okay, but fungi do the same thing that we do. They make digestive enzymes that can break down the organic material that other organisms are made of, either as a decomposer, decomposing something that's already dead, humus, plants, animals, whatever, okay? Or they could be parasitic, which means they actually live on a living organism and are actively eating that living organism. So if you've ever seen like a bracket fungus that grows on the trunk of a tree, okay, that's a parasitic fungus. Okay, it has got its hyphae and its mycelium inside the tree. and it's actively stealing energy from the sap that the tree is making, the sugary sap. Okay, it's taking energy from that. Okay, it's breaking down parts of the tree for food. Okay, secreting digestive enzymes. What's different is we make those enzymes, and our digestion is internal. We eat stuff that goes into our digestive tract, it's digested inside our body. A fungus uses external digestion. So the little hyphae, if my finger is the hyphae, okay, it secretes the enzymes and they're outside of the hyphae. So it dissolves and digests the substrate, whatever it is, okay, around it, and then absorbs it into the hyphae. So the digestion actually happens outside of the fungus, and then it absorbs it from there. All right, that's how athlete's foot works. Okay, so if you had athlete's foot, okay, it's itchy. Right? It is actually a fungus that is growing on your foot. It's putting its little hyphae in between your cells. It's secreting digestive enzymes that are eating your cells. It's chemically breaking them down and is digesting you. Okay? That's why it's itchy. It's itchy because it's actually destroying your cells and eating them. You can rock yourself to sleep tonight with that thought. Okay, but that's how fungus work. Okay, if you have a fungus, okay, or there's a fungus like in your yard, if anyone's like had the fairy ring, you know, where you get that dead ring of dead grass, that's a fungus. Okay, that fungus is actually growing and eating the roots of the grass. Okay, it can do those kinds of things. So that sort of makes sense. Okay, if you have like you know, moldy bread, okay, or something that's growing mold in your fridge, same deal. It's digesting it externally and absorbing it. All right, we will leave it there, guys, for today. Make sure if you want to start at noon, show up at noon, okay, to start writing your exam. Otherwise, show up, start a class. Okay. Pencils, plural, calculator, and good batteries, periodic table with not too many notes to self on. Okay, have a good weekend, guys, but not too good.